Anybody want to try to explain the difference between them and why you might hypothesize one might be preferable to the other?
I would just think that the semi-conservative is probably more logical, just there's less steps involved. Yeah. Just because it can be explained how it works.
What's that? Yeah, go for it.
Right?
Yeah.
So they just split, like the first DNA strand splits in half, or the original one, and then they just form two other ones like off the template, and they just create those two new strands. So like they're split half old, half new. The conservative is like just more steps. The old one splits as a template, and then the new halves put together another one, and the old one's about the other two. So just two steps.
So this one seems like the most simple, and then because many things, if you have to choose between a simple and very complex one, you might say, well, the simple one is probably the more likely approach. Anybody want to take up the torch for conservative replication?
Not being here, I'm just taking a wild guess. The more complicated one is always mostly best because you need more samples when doing experiments. So the more steps you have to go through, the more results you get. So you have more to compare to.
Okay, so maybe in an experiment, when you're setting up the experiment, maybe taking a more complex approach to designing it might give you the opportunity to get better results. But in this case, we're not talking about the experiment itself. We're talking just about sort of different ways that we think living systems actually do it. So they're just hypotheses about how it might happen, as opposed to sort of methods we're going to use to figure out which one it is. Yeah?
Well, so I would argue that the original strand divides, and then they create a new strand. They divide again. The originals are together, and the copies are together. You could say that the original would probably make it stronger, being the original, but there might be the possibility for problems with the replicating ones going back together.
Yeah, so one thing, okay, I'm not sure I totally understood the last point you had to make, but what I was hearing in the first part of it was by putting together your original double helix at the end of copying, you preserve an unaltered, error-free copy. All right? And then you put together the copy, the two copies, but your template, your original template for both sides, remains intact. Does that argument make sense to everybody? You see why you could argue either one of these as being a logical possibility? Yeah.
Well, I can see the conservative as being logical in one sense because we have an original cell that we're starting with, and as you said, an original template by which we're going to create the new strand of DNA from. And it says coming from an original cell with its original DNA, you would potentially make the argument that the original DNA should stay with the original cell because you wouldn't want it to be altered, one, and two, that presents all the heritable information for that specific cell that engineers and operates that specific cell.
Yeah.
And then the new cell is going to take on the new information whether or not it does have errors, which most likely it does. It is extremely complex, but that new strand of DNA has been formatted and fit to work within that new cell. And so because it's specifically made for that one, it should go with that one, the original. Go with the original.
Okay, so maybe there's sort of adaptation happening within, mutation and adaptation happening within a cell line?
Maybe.
Possibly. So to be clear, does everybody see how if you do semi-conservative replication and you go from one cell with its DNA to two cells with the same DNA, you have the possibility that one of these cells or both might end up having a mutation introduced, because copying is just an enzymatic process where you add letters that fit, and every once in a while, one in many millions, you make an error. All right, and so by keeping together the original, you have an error-free template where you can generate new copies to fit these different environments, but you have one original. Okay, I think we were all set up now to really be excited to figure out if it is conservative or if it's semi-conservative. We'll just ignore dispersive. But actually, in the experiment we're going to do, it's not until the end of the experiment that we actually exclude dispersive as a possibility. So you have to keep it in your mind. And remember, as an experimentalist, you come up with all the hypotheses that could possibly, you want to come up with hypotheses that will be testable. And so all of these possibilities need to be able to be excluded in our experiment for us to conclude that it's most likely one of these. Okay, so let's get started. So in order to explain this experiment, and this is the last experiment on your sheet here, I'm going to kind of do like I did for the experiment on the front. This one that had the phosphorus 32 and sulfur 35. And I'm going to take some time and explain the experiment, and then I'm going to have you guys with your groups discuss the results and tell me what you think it is. Okay? Does that sound good? So it'll probably take about 10 minutes to sort of set up the experiment, and then you'll look at the data here, which I think at the moment probably doesn't mean much. And hopefully you'll be able to interpret it correctly. So in order to do this, I have to tell you about a few things. I have to tell you about centrifuges, and I have to tell you about gradients. So what's a centrifuge? Yeah, so it spins something that spins really, really fast and separates something from something else. Do you guys employ centrifuges in your daily life? You do, in fact, employ centrifuge. Well, either you do or your parents do when they wash your clothes for you. So you put it into the washing machine, you put a bunch of water in there, you shake it up, and then you spin it, and all the water separates out from the clothes and moves through those holes in the side of the machine. So you really use a centrifuge whenever you wash your clothes. So a centrifuge is something that spins really, really quickly. And I need to give you sort of the setup for the experiment by explaining next what a gradient is. And I want you to envision a tube. And into this tube, I'm going to have a hose. It's coming from a really, really very salty solution, and another hose is coming from pure water. Do you have this in your mind? So if I start filling the tube from these hoses, and as I fill, I start sort of pulling these tubes up out of it, and change the relative amounts that are coming into the tube from the two sides. That is, if I start just filling with really, really salty solution, the bottom of this is going to be incredibly salty. But then as I start filling, I add a little bit of water, and then keep adding more and more water, so it becomes a larger and larger proportion of what's being added to the tube. What will happen is I'll have this tube that gets salty, less salty, as it goes up to the top. So the bottom here is going to be really salty, and the top here is going to be very little salt. And that's a gradient, all right? It's a gradient in saltiness. And one of the really, really magical and weird things is that when you take a molecule and you load it onto the top of one of these tubes, and you spin it in a centrifuge, it actually moves down the gradient to the point where it has the same density, in essence, as the salt around it.